Hello, my name is Rich Whitworth. I'm editor of Biofarm International, and I'm here today with Jörg Zimmermann, who's the Director of Process Development and Implementation at Veta. Welcome, Jörg, and thank you for joining me today.
Hello, Rick. My pleasure.
So maybe first of all, you can give us a little bit of information about your own background and experience in the pharma industry.
Yes, definitely. So by profession, I'm a pharmacist. I'm a registered pharmacist here in Germany, and I did most of my studies in Germany, but I also did some time at the Welsh School of Pharmacy in Cardiff.
Oh, really?
Yeah. And I actually then started as a pre-veg pharma student at Veta Pharma, which at that time was a company of less than 500 people, if I remember correctly. So I started in a production environment as an assistant head of production. Did a lot of development work already then within production because we didn't have a separate department for it. And in 2001, I moved into one of the new production sites that we were just building up in Germany. So I was heading that site and building it up from one production line up to 5 production lines. And I'm happy to say that that site is fully operational and producing lots of pre-filled syringes every year. And when the company asked me to do something new and move into process development and process implementation, I took that opportunity to be able to develop processes that are commercially viable and make the most sense for the customer in terms of quality and cost, of course. And that's sort of I started in '94 at Veta, so it's a good 17, 18 years now that I've been with the company and growing with it. And it's I have to say it's still a lot of fun doing it because there's so many interesting things coming our way in injectable products and biologics.
And maybe how have things changed over that long period of time, would you say? Do you think there have been a huge number of sort of technological advances, or do you think it's been a fairly steady progress?
Well, I think in the first place, the pharmaceutical industry is a very conservative industry, so innovations sometimes take a little longer to come through versus maybe the food industry or automobile industry. But at the same time, in our special field in injectable drugs, with the advent of all the biotech products going from recombinant proteins through to monoclonal antibodies and the more innovative designs like microparticles that deliver the product over time and fusion proteins and products that combine an albumin with a therapeutic protein, there's a lot of advances that were made on that. And also in terms of what the regulators require, the standards are being pushed higher and higher and even higher because essentially we're doing products that are injected directly into people's bodies, circumventing all the natural defense mechanisms. So we've got to be sure that they're safe and sterile. So in that respect, I would say the industry has been advancing at a very rapid pace. And sometimes when I look at the standards that we were just using 5 or 6 years ago, they just don't hold anymore.
Right, and having had the experience of that sort of process and moving through with the changes, you've also probably picked up some important knowledge along the way that you can certainly help you in the position of process development.
Oh, absolutely. There's a lot of the things that you've been through and the parallels that you can draw when a new problem comes up. So you can go back and think, oh, we had something similar back in blah blah, and then find out what the best solution is for that. So sometimes learning the hard way, but that's how it is.
Well, that's often the not the favourite way, but the best way. We probably say.
Yeah. Yes.
And we had a recent article by Paul Nells, actually, also at Veta, in our drug development basic training series. And the article basically summed up that when at the discovery stage, very little thought is given to the later development or commercialization efforts that need to be applied to a compound. So maybe you can give us a sort of brief roundup of some of those important considerations.
Mm-hmm. Yeah, there's a couple of good examples that I can give you here. For example, let's think of a company that has a very novel delivery device that is I don't know, something like a patch pump or an auto-injector or something like that, which is technologically very advanced, but it still needs some ammunition to get the drug in. And that ammunitions got to be filled in an aseptic manner to get to a sterile product. So you're looking at a combination of a delivery device with a cartridge or a syringe. And just recently I had an example of a company that was very far advanced with their device because they thought, oh, the cartridge, that can't be the problem. And then when we looked at it in more detail, we saw that the cartridge that they had looked at was not manufacturable in an aseptic way. And to get that implemented, we were looking at, unfortunately, quite a lot of money and a long lead time to get it going. And that was sort of it was sort of a dead end for that development that, well, they didn't think about it in the beginning. So in essence, what I'm saying with this example is a lot of times you take things for granted or you think that they're normal and natural and already solved, but you really have to look far ahead to in the overall development to avoid those pitfalls. So
right.
It's like the point where you say, yeah, we'll make it up on the way. You'll end up going a lot of ways before finding the right one.
Talking about thinking further ahead, the FDA's sort of advised that biologic formulations in pre-filled syringes now need to be considered as a combination product, or will in the near future. So there's more regulatory focus as well on establishing, formulation compatibility with the pre-filled syringe components. So maybe you could sort of tell us your kind of viewpoint on that as well.
Yes, absolutely. The traditional viewpoint was that a pre-filled syringe was just a packaging component and that it was just a drug and not a delivery device. But in essence it is, because the syringe functionality is the basis of the delivery. So you have to make sure that a syringe or cartridge is working properly, and in this case the combination product and design requirements that come with it are essential. It's something that has been done all the way through, but it's never been labeled that way. And maybe with the standards that are used there, it's being considered much more in that sense. And I think it's definitely the right way to go, to consider such a combination product as a combination product. To add one example to that, it's in a combination of diluent syringe with a vial, a live vial, and an adapter that goes with it. So a real-life example, we were producing something for the market in a diluent syringe for a customer that was doing the live vials themselves, and they were using an adapter. And all of a sudden, we were getting complaints from the customer, and I think also from the market, that it didn't work anymore, that they couldn't do the reconstitution properly. So we looked at it in more detail, and what we found was that the stopper that was used in the vial had been changed in design. So they were doing an optimization of that design as part of the lyophilization process, but they didn't consider that that was going to have an impact on the adapter syringe vial combination. And all of a sudden we couldn't get the product reconstituted in the way that we had done it before. So it was a quick fix that needed to be done, because it was already a marketed product and we had to make sure that the patients were getting their drug. So in this case, design controls, if you apply them properly, you can get those problems sorted before they hit the market.
And do you think that the regulatory requirement to start considering that earlier on will help some of these companies to push through these particular pitfalls, or do you think it's inherent in the process that a lot of these companies are actually fairly young and maybe inexperienced with drug development, so they probably need to work with other people in the industry that have got more experience?
I think it will help, but again, experience is invaluable in this respect. And a lot of the things with the experience, you can tell them straight away by just looking at it. And other people might think, oh, this is easy, we can do it, no problem. And then you say, oh, have you thought about this, and have you thought about that? And how about the secure excipients, for example? How many sources do you have? Will you rely just on one excipient source for a critical component? And what if that supplier is no longer able to supply? So it's a lot to do with playing devil's advocate as well in the development, and doing a proper risk analysis and risk management.
Yeah, certainly. And you've already touched on this, I guess, but as you already said, some of the highly sensitive drugs, especially biopharmaceutical ones, are actually requiring more optimally designed drug delivery systems. So perhaps you could run through a few sort of technical challenges that companies may face through drug development. I know you've already given us a couple of examples, but maybe you could dwell a little bit more on that.
Yeah, I think one trend that we see at the moment is the trend towards smaller injection volumes and higher concentrations in products. So you're looking at high dose, maybe 200 mg per mil protein solutions, maybe pegylated monoclonal antibodies, maybe other dosage forms that have a lot of product in them in a very small volume, and the high concentration also leads to higher viscosity and challenges on that. So there's people working on, as I said, pump designs that allow to dose higher concentrated products over a longer period of time, or dosing into the central nervous system and things like that. So those are all the latest and greatest developments that we see, and they all come with all kinds of new challenges. So if you look at such a high concentrated protein solution, it is viscous, it'll dry quickly, and you're filling in laminar airflow with a 0.45 meters per second airflow velocity at the point of filling. So if you have the filling line stopped for just a minute or two, sometimes you can almost see product building up on that filling needle. So what do you do with that? We've developed special needle tip designs and procedures to avoid that, because you don't want to get crystallized protein into your liquid solution. Or in the worst case, get the filling needles clogged altogether, and you have to stop the run and start all over again. So these are some of the things that are coming up. Also, with these high concentrated products, you're looking at how can the dosing into the container be done in the first place. The product may be highly shear sensitive. Can you dose it with a rotary piston or rotary vane pump, or maybe you have to go to a peristaltic pump or a membrane pump to make it more gentle? And there's also some really interesting examples that we've gone through in the past. Again, unfortunately, a few years back, learning the hard way in real-life production for the market. And the lesson learned that we have from that is that we now have a process development lab that I'm running, where we can find out about those things before we hit the commercial filling line. So we will be doing dosing studies with limited amount of product solution to find out, do we get product aggregation or particle formation in the dosing of the solution, and what does it take to get out of it, like selecting the right pump system, selecting the right needle tip designs for the filling needles, and so on and so forth. So it's taking the lessons learned and working them into preventive actions for further process development.
And I suppose in many ways, because of the nature of biological products, each drug development is a very much a case-by-case basis, because different biologics and different proteins interact in different ways and can aggregate under different conditions and so on. So it really is, there are no hard and fast rules.
That's right, yes. As I said, you can draw on the experiences that you've already have, but it's still something that you still have to go through case by case. So in terms of compatibility with silicone oil that we still require for lubrication of the syringes, one product might be very sensitive to it and lead to aggregation or turbidity in the solution, and other products will be completely oblivious to that and stay a clear solution. Or if you look at, just another example, if you look at a multi-dose cartridge going into a delivery device, those multi-dose products, of course, need a preservative, because once you've pierced the septum of the cartridge, you have breached the integrity of the unit and you could get germs into it. So you'll need some sort of preservative like metacresol or chlorocresol or something else, benzyl alcohol. Now those are organic solvents, and you have to look at the interaction that you get with the silicone that you're using for the functionality of the cartridge. So depending on which preservative you use there, you might be ending up extracting a lot more from the glass surface with one versus the other. So it's again something about shelf life that's got to be considered. And that's something, I was bringing that up to a customer the other day, and they said, oh, we haven't thought about that. That's a new point. We should have thought about that 3 years ago. So,
I guess that happens quite a lot. That because of your experience and the fact that you're working with a lot of different companies, that some things that seem fairly obvious to you, perhaps particularly to a new company, can be the first time of hearing. So
yeah, and for us, it's more the point of not taking what you know for granted, but making sure that you always think about it and not think it's natural and everybody knows about it.
Right, sure.
Always go back and recheck your assumptions.
Right.
Even if it's stating the obvious.
Yes, sometimes stating the obvious is necessary. Now, you've mentioned siliconization there, and it's clearly one of the big challenges, and perhaps something that a lot of companies don't consider. Maybe you could give us a bit more sort of technical background as to why that is such a challenge.
Well, yeah, if you think about it, it is not rocket science because it's some basics behind it. But at the same time, as a lot of people don't know too much about it, if something goes wrong, they'll say, it's got to be the siliconization. and then you have a hard time proving that that is not the case and that there's 1,000 other factors influencing it. Well, let's go back. Why do we need the silicon in the first place? It's really to guarantee the functionality of the syringe, which means that you can still push the plunger in the syringe at a reasonable force to get to the dose without it stopping or without it moving too fast and without having too much silicon that it gets maybe liquid oil droplets into the solution. So the key is to have as little as possible and as much as needed to make the syringe work. And ideally to have it evenly distributed throughout the glass barrel so that the syringe will move, the stopper will move smoothly from the beginning to the end through the. We've done a lot of work on that over the last 10 years, I would say, because how do you get the silicon in the syringe? Well, you've got to apply it in some way, and traditionally with oil that had been done in a swab type method, which wasn't giving too good results. But giving not too good results still gave you a functional syringe. But once you put it into an auto-injector where you don't have anybody regulating the force to push the plunger, gave us problems. So, and that's the difference. When you have the syringe and you apply it manually, a human being can alter the force. If you have an auto-injector, there's a fixed spring force, and
of course, yeah.
It either delivers or it doesn't. So it's a lot less forgiving as a system. So with that in mind, we developed the spraying processes for silicon oil, dynamic spraying processes, where we deliver the smallest silicon amount all the way from the top of the syringe to the finger flange of the syringe. At the same time, we also had to develop the right analytical methods to prove that we have that even distribution of the silicon. Because it's really not much. You're looking at 0.1 to 0.6 milligrams per glass barrel. It's really minute amounts, and you still have to make them visible somehow. So a lot of work went into that analytical method development as well. And then to have it installed on all the filling lines was the next thing. The industry overall has learned a lot on that, and a lot of people have jumped on that, including the primary packaging suppliers, and things that were rocket science like 5 or 6 years ago are now commonplace. And that again is a technological development that everybody's profiting from.
And I guess for biological products, the trend seems to be, if not already, that a lot of these medications will be self-administered. So it stands to reason, I guess, that auto-injectors will become more widely used as well. So more and more companies are going to be facing these issues, I think it's fair to say.
Absolutely. So we were having a very extensive optimization project with a customer where we had launched a prefilled syringe auto-injector pen combination, and when we had complaints coming back from the market and also some problems that we were seeing in production. In the end, what always surprises me is you look at an auto-injector and you think this has to be self-explanatory, but then in comes the user and we see all the problems and all the things that can go wrong with it. A lot of that's something that people maybe also underestimate. They say, oh, it's an auto-injector, it's got to be self-explanatory. You bring it to the market, patients will be fine with it. I know how it works. They will know how it works too. But it's a huge educational effort that the companies go through these days. Some companies, for smaller products or specialty products, they might even know their patients by name if it's orphan drugs, so they can target their audience and really go to those patients directly. But other than that, even for the bigger products, they will launch test kits or practice kits for the patients to try the auto-injector before they start the first injection. They'll have the sessions with the doctors before they do it the first time. They will have educational videos and web pages and everything. So there's a lot of effort being put into that. And you see that once the introduction of a product is done, you will get a bigger number of complaints in the beginning, but that'll dwindle down quite quickly as the patient population picks up on it.
Finally, maybe you could give some advice on what small or startup biotech companies should be doing, I guess, to fully prepare themselves for a smooth drug development. Maybe some tips and advice just to get people into the right frame of mind.
Mm-hmm. And as always, the things that you don't know, you underestimate. So the general advice is to really think it through, and as I said before, not go in thinking, oh, we'll make it up on the way, and then end up finding all kinds of problems. So it's really in the development, when you develop your timelines. I know the companies are under a lot of pressure to get product to the market and to show the shareholders in the companies that they're advancing quickly. But a lot of times it's a better approach to get to maybe a little more conservative but realistic timeline, and then not to have to extend it every other month. It's a little bit like Boeing launching the 787, where they had, I don't know, a total of 3 years delays, but they announced it sort of in 3 to 6 months intervals. Oh, we're not ready yet. Oh, we're not ready yet. So it's that kind of thinking. So a lot of times you really have to go in and play devil's advocate and have to question all the things that you assume to be normal and really do the risk analysis. So the process FMEAs like failure mode and effect analysis, it's a painful task because you're thinking of all the things that could go wrong, and do I have the right measures in place to avoid them, and if not, do I have to put in more measures? So that's something that can take a group of experts a day, 2 days, maybe 3 days to go through just the filling process from start to finish. And then once you've gathered some real experience with the process, you have to go through it again and check, oh, is everything in place? Is the process safe and secure now? So it is a painful exercise, but at the same time, it makes you think a little further than just, oh yeah, it'll be fine. So you will find the things that could go wrong, and you make sure that you avoid those. So that's sort of the things that I'm always trying to push. If you then find out in the timeline, oh, this was too conservative, we're moving ahead much more quickly, then that's fine. That's a gain for everybody. But I'd rather go that way than the other.
Okay, now, are there any comments you'd like to add at the end? Anything else you'd like to tell our listeners?
Well, I think it's really, as always, try to level with your partners and try to listen in both ways, because I mentioned before orphan drugs. The game that orphan drugs are being played at is different from traditional pharma. So always listen to your customer, listen to your partners, try to be on an even level and learn from each other. It's a team approach, as always.
Well, thank you so much for your time, Jörg. It's been a pleasure talking to you, and we look forward to hearing more from you at some point in the future. Thank you.
Thank you very much from my side as well.